Cell respiration.
Cellular respiration is a series of reactions that takes glucose and uses it to synthesize ATP from ADP and an inorganic phosphate. This takes place in the cytoplasm of the cell and the mitochondria of the cell.
Glycolysis is the first step of cell respiration. It is the process in which one glucose molecule is broken down in a series of reactions in the cell's cytoplasm, which are all catalyzed. This creates 2-pyruvate, releasing energy. 2 ATP molecules are used up as an energy input. A glucose 6-phosphate is created when the first ATP attaches a phosphate group to one end of the glucose molecule. Glucose 6-phosphate is rearranged into its isomer fructose 6-phosphate. Fructose 1,6-biphosphate is created when the second ATP attaches a phosphate group to another end of the fructose 6-phosphate. A phosphate is transferred to the 6-carbon backbone by ATP. The backbone is split, forming glyceride 3-phosphate and dihydroxyacetone. DHAP is then converted into G3P. This creates an energy release. G3P gives up 1 hydrogen and 2 electrons to NAD+, which reduces to NADH. At the same time, the G3P combines with inorganic phosphate, which results in unstable molecule called 1,3-biphosphoglycerate. ATP is formed when 1,3-BPG gives up a phosphate group to ADP, resulting in 3-phosphoglycerate. The molecule of 3-phosphoglycerate rearranged to its structural isomer 2-phosphoglycerate. 2-phosphoglycerate gives up 1 proton of hydrogen and 1 hydroxide ion, which combine to form water. The resulting molecule is phosphoenolpyruvate. This molecule is very unstable and breaks down. This releases ADP, which forms ATP. The original glucose molecule has been broken down to 2 pyruvate molecules, each with a 3-carbon backbone.
Pyruvate oxidation. 2 pyruvate molecules enter the mitochondrial matrix via a membrane carrier to help it pass the outer and inner membrane. Coenzyme A binds to the acetyl group on the pyruvate, allowing it to be oxidized, while NAD+ undergoes a reduction, making NADH. The carboxyl group that remains is waste and is removed as CO2. This results in an acetyl-CoA. The acetyl-CoA enters the KRB cycle, where energy is needed to continue aerobic respiration.
The KRB cycle. The acetyl group from acetyl-CoA joins alkoxylacetate to make citrate. Citrate becomes its isomer, isocitrate. Isocitrate loses CO2 and 2 hydrogens and becomes alpha-ketoglutarate. 2 hydrogens are lost, and this reduces NAD+ to NADH. Alpha-ketoglutarate binds to CoA and is oxidized by NAD+ and becomes succinyl-CoA, while CO2 is removed. Succinyl-CoA loses a CoA, forming succinate. The CoA is displaced by a phosphate. GDP is phosphoglycerated, making GTP. The phosphate then turns ADP into ATP. FAD from the matrix oxidizes succinate, making FADH2. Succinate is now fumarate. Water is added, making malate. Malate is reduced with NAD+ to make NADH and oxalacetate.
Electron transport chain. The proteins involved in ETC are lined up in order from lowest electronegativity to the protein with the highest electronegativity. The electron's energy is passed along the membrane. UQ, a mobile electron carrier, carries electrons from NADH dehydrogenase to cytochrome BC1, while Site C carries electrons from cytochrome BC1 to cytochrome oxidase. FADH2 does the same but starts at the succinate. This forms less ATP. Two electrons leave cytochrome oxidase and each combine with an oxygen, the last electron acceptor, and making hydrogen, forming water. Oxygen is used because it has a powerful enough electronegativity to take the electrons from the last components of ETC.